Pyruvate dehydrogenase deficiency is a serious problem for all cells with mitochondria, since it prevents the products of glycolysis from entering the TCA cycle and the subsequent electron transport chain for aerobic respiration. The subsequent backup of substrates, including pyruvate and alanine, increases the amount of anaerobic fermentation, causing lactic acidosis. This deficiency can be congenital or acquired, which is most common from B1 deficiency in alcoholics. Clinically, this deficiency manifests as neurologic deficits. It's treated by increasing intake of ketogenic nutrients, such as a high-fat diet or the ketogenic amino acids, which are lysine and leucine. These nutrients do not have to go through a pyruvate intermediate to enter the TCA cycle. Pyruvate is an important intersection point for several metabolic pathways. First, pyruvate can be converted to alanine in the liver via alanine transaminase, or ALT. This allows amino groups to be transferred from the muscle to the liver. Second, pyruvate can be converted to oxaloacetate via pyruvate carboxylase to either replenish the TCA cycle or for use in gluconeogenesis. Third, as I've shown before, pyruvate can link glycolysis to the TCA cycle when converted to acetyl coenzyme A via pyruvate dehydrogenase. And fourth, pyruvate itself can be an endpoint to anaerobic glycolysis when it's converted to lactate via lactate dehydrogenase. This reaction provides additional NAD+, so that glycolysis can continue to produce ATP, and anaerobic ATP production is the major pathway in red blood cells, leukocytes, the kidney medulla, the lens of the eye, testes, and the cornea. The TCA cycle, or the Krebs cycle, takes place in the mitochondria and functions to produce high-energy electron carriers, such as NADH and FADH2, for further ATP generation in the electron transport chain. This completes the metabolism of glucose. This diagram shows the major steps in the TCA cycle. Pyruvate is the main substrate or input to the cycle and is converted to acetyl-CoA, which produces one NADH and one CO2. Proteins and fats can also enter the cycle after being converted to acetyl-CoA. The net cycle produces 3 NADH, 1 FADH2, 2 carbon dioxide, and 1 GTP per cycle, which amounts to 12 ATP per acetyl coenzyme A, since each NADH allows the production of 3 ATP and each FADH2 allows the production of 2 ATP. Since each glucose can produce 2 acetyl coenzyme A, this means you can get 24 ATP per glucose. The TCA cycle is controlled at 3 major irreversible steps, which are shown in bold in this diagram. Citrate synthase, which can be inhibited by ATP, combines acetyl-CoA with oxaloacetate to form citrate. Isocitrate dehydrogenase, which is also inhibited by ATP as well as NAD and activated by ADP, converts isocitrate to alpha-ketoglutarate. And finally, alpha-ketoglutarate dehydrogenase converts alpha-ketoglutarate to succinyl coenzyme A. Therefore, it makes sense that succinyl coenzyme A inhibits this reaction, since if you already have a lot of succinyl coenzyme A, you don't want to keep making it. Also, again, ATP and NADH inhibit this, since these molecules are both only found in high energy states, in which you don't need to produce more energy. As I mentioned before, the alpha-ketoglutarate dehydrogenase complex requires the same cofactors as the pyruvate dehydrogenase complex, which are vitamins B1, B2, B3, B5, and lipoic acid. A useful mnemonic for remembering the molecules in the TCA cycle is citrate is Krebs' starting substrate for making oxaloacetate. In the electron transport chain, NADH enters at complex 1 and FADH2 enters at complex 2, which is also succinate dehydrogenase. As electrons are passed through the complexes, they create a proton gradient, which is used to drive the production of ATP. Complex 4 is also known as cytochrome C oxidase, and this enzyme transfers electrons to the final electron acceptor, which is oxygen, to make molecular water. Complex 5 is also known as ATP synthase. It produces 3 ATP for every NADH to enter, and 2 ATP for every FADH2. Since FADH2 starts at a later point, and therefore is not able to pump as many protons across the membrane. Electron transport inhibitors such as rotenone, cyanide, antimycin A, and carbon monoxide directly inhibit electron transport from complex to complex. This prevents the formation of a proton gradient and therefore the synthesis of ATP by ATP synthase. Rotenone inhibits complex 1, antimycin A inhibits complex 3, and cyanide inhibits complex 4. Oligomycin is an ATPase inhibitor, and therefore inhibits complex 5. This allows the proton gradient to continue to increase without any ATP formation. As electrons build up, electron transport stops. Uncoupling agents, such as 2,4-dinitrophenol, aspirin, and the protein thermogenin, which is produced in brown fat, increase the permeability of the inner membrane, which allows protons to flow through. This results in increased oxygen consumption, since an increase in electron transport attempts to compensate for the membrane permeability. Still, ATP cannot be synthesized without the proton gradient, and the increase in electron transport is instead dissipated as heat. This is why aspirin overdoses can cause fevers, and why babies can stay warm since they have lots of brown fat. Gluconeogenesis, which is shown here, is basically the reverse reaction of glycolysis. It begins with pyruvate and ends with glucose. There are 4 irreversible enzymes of gluconeogenesis. The first is pyruvate carboxylase, which converts pyruvate to oxaloacetate. This occurs in the mitochondria and requires biotin and ATP and is activated by acetyl-CoA. That reaction is shown here at the bottom of the diagram. Next, PEP carboxykinase converts oxaloacetate to phosphoenolpyruvate, or PEP, in the cytosol, which requires GTP as a source of the phosphate. Fructose 1,6-bisphosphatase is the third irreversible enzyme, which converts fructose 1,6-bisphosphate to fructose 6-phosphate. And finally, glucose 6-phosphatase removes the phosphate from glucose 6-phosphate to form glucose. This last step occurs in the endoplasmic reticulum and does not occur in muscles since they lack this enzyme. Therefore, muscles cannot participate in gluconeogenesis. Glucose 6-phosphatase is also the enzyme that's mutated in von Guericke's disease, which we'll talk about towards the end of the lecture. Gluconeogenesis occurs primarily in the liver and creates glucose from glycolysis, fatty acid, or TCA cycle intermediates. A deficiency of gluconeogenic enzymes can cause hypoglycemia. It should come as no surprise that a deficiency of gluconeogenic enzymes can cause hypoglycemia. Odd-chain fatty acids can serve as a glucose source because they yield propionyl coenzyme A, which can enter the TCA cycle as succinyl coenzyme A. Even-chain fatty acids, however, cannot be a glucose source, since they only yield acetyl coenzyme A equivalents. The hexose monophosphate shunt, also called the pentose phosphate pathway, is shown here. Its purpose is to synthesize NADPH when glucose 6-phosphate is abundantly available. The NADPH can subsequently be used in reductive and anabolic reactions, such as in glutathione reduction in red blood cells and cholesterol synthesis. Low NADPH can cause hemolytic anemia due to accumulation of oxidative damage. If the cell needs lots of NADPH, it can make this pathway into a cycle by converting fructose 6-phosphate into glucose 6-phosphate. The HMP shunt also synthesizes ribose, which can be used for nucleic acid synthesis and glycolytic intermediates. The pathway consists of an oxidative, irreversible phase, followed by a non-oxidative, reversible phase, both of which take place in the cytoplasm of lactating mammary glands, liver, adrenal cortex, and red blood cells. The transketalases require vitamin B1 and are often used to test for vitamin B1 deficiency. The respiratory burst, or oxidative burst, involves the release of reactive oxygen intermediates by immune cells such as neutrophils and macrophages, which kill invading bacteria or fungi. The first step involves the activation of membrane-bound NADPH oxidase, which converts molecular oxygen into superoxide radicals. Superoxide dismutase then converts the superoxide to hydrogen peroxide, which can then either be chlorinated by myeloperoxidase to make hypochlorite or bleach, or be detoxified by catalase to make water. Since bleach can be used to kill bacteria, this is a useful pathway for both neutrophils and macrophages. However, hydrogen peroxide is also toxic to the host cells, and therefore the glutathione pathway is used to neutralize it. So what happens if a patient is missing this first enzyme, NADPH oxidase? Well, these patients develop chronic granulomatous disease because they cannot produce superoxide radicals or hypochlorite to kill bacteria. They can still use hydrogen peroxide produced by invading organisms to produce hypochlorite. However, they have increased risk of infection from catalase-positive species, such as Staph aureus and Aspergillus, because these species will use their own catalase to convert the hydrogen peroxide into water. Glucose 6-phosphate dehydrogenase is involved in both the HMP shunt and the respiratory burst. It's the key enzyme that regenerates NADPH, which is a very important reducing agent that is oxidized so that glutathione can remain reduced. Glutathione, in turn, can detoxify free radicals and peroxides in red blood cells. If red blood cells are not able to produce NADPH, this can cause hemolytic anemia since they have no defense against oxidative stress. Favabeans, sulfonamides, primaquine, and some antituberculosis drugs can all produce reactive oxygen species, which normally are neutralized by glutathione, but patients who are deficient in G6PD can't produce enough NADPH to reduce glutathione, and therefore can't neutralize dangerous oxide radicals. G6PD deficiency is an X-linked recessive disorder and is actually the most common human enzyme deficiency, with the highest prevalence among blacks. This may be because it provides some resistance against malaria. One common sign of G6PD deficiency is the presence of Heinz bodies on a peripheral blood smear. These red dots that appear in red blood cells are oxidized hemoglobin that can precipitate. Bite cells can result when these Heinz bodies, in addition to part of the red blood cell membrane, are bitten out by macrophages. These are both covered in more depth in the Hem/Onc chapter. Fructose metabolism is important because it converts dietary monosaccharide fructose into a substrate for glycolysis and energy production. As you can see in this diagram we covered a few minutes ago, fructose can enter glycolysis partway through the pathway. Normally, it's converted to fructose 1-phosphate by fructokinase in the liver. The enzyme aldolase B then converts fructose 1-phosphate to dihydroxyacetone phosphate and glyceraldehyde. Triose kinase can then combine these two to make glyceraldehyde 3-phosphate, which then enters glycolysis. Essential fructosuria is an autosomal recessive defect in fructokinase. It's a benign asymptomatic condition, since fructose never enters the cells and is just washed out in the urine. However, fructose intolerance is a more serious autosomal recessive disorder of aldolase B. Without aldolase B, fructose 1-phosphate accumulates. And since this was made inside the cell and can't leave it because it has a phosphate group attached, fructose 1-phosphate will build up inside the cell. This consumes lots of the available phosphate from the cell, which then inhibits glycogenolysis and gluconeogenesis. Decreased ability to synthesize glucose results in hypoglycemia, and patients also demonstrate jaundice, cirrhosis, and vomiting, since the accumulation of fructose 1-phosphate becomes toxic to the liver. The treatment for this is to decrease the intake of both fructose and sucrose, since sucrose is composed of both glucose and fructose. In general, disorders of galactose metabolism, which we'll discuss next, are more serious. In galactose metabolism, lactose and galactose are converted to a more usable form that can enter glycolysis. It occurs in the kidney, liver, and brain. Normally, dietary lactose is broken down into galactose and glucose. Galactose is then phosphorylated by galactokinase to galactose 1-phosphate, which is converted by uridyl transferase to glucose 1-phosphate. This, of course, can be used for glycolysis or gluconeogenesis. Galactokinase deficiency is an autosomal recessive deficiency of galactokinase. This prevents the conversion of galactose to galactose 1-phosphate and instead causes it to be converted to an alcohol form called galactitol. It's usually a mild condition, but the galactitol accumulation inside cells can cause galactosemia, infantile cataracts, and failure to track objects or develop a social smile. Classic galactosemia is a more serious autosomal recessive deficiency of galactose 1-phosphate. Classic galactosemia is a more serious autosomal recessive deficiency of galactose 1-phosphate uridyl transferase. This causes both galactose 1-phosphate and galactitol to accumulate inside cells, so it causes some of the same signs and symptoms of galactokinase deficiency, such as infantile cataracts, as well as some new ones from the galactose 1-phosphate accumulation, such as jaundice and hepatomegaly. The treatment is to not eat anything with either galactose or lactose.